core feature of Auricle. So as we know, at Auricle, we provide live classes, recorded sessions, notes, MCQ. So live classes are to elevate the learning and where knowledge meets instruction. Recorded sessions are provided for the convenience of the student, where for all the first year student will have the access of all the subjects anytime, anywhere through the recorded sessions. And also, we provide ultimate comprehensive books also, that is ABME based question answers, MCQs and full zone for self-assessment. And also MCQs are there from beginner level to expert level, which contain standard multiple choice questions. Then also in Auricle, the student can book a slot for personalized counseling session in which the student will have the session with the counselor. Then we also have some subscription plans. So as we can see, the subscription plan starting from the basic to premium, right? So basic plan is of 3 months, standard plan is of 6 months, and premium plan is of 12 months. So the premium plan costs around 50,000, and all the services which is provided by Auricle, it includes like pre-recorded sessions, live class, live doubt clarification, one-to-one personalized guidance, notes, quick revision, examination module, and also students can use my code for 20% instant discount. The code is Anchal20. Okay, so as we have started the internal exam marathon series, so the core feature that we provide in this rapid revision for all the important topics and also for volatile fact revision, quick revision, and multiple choice questions will be also covered, and also we assure top-notch grades in exam. So these are the topics that we're going to cover, starting from fatigue in skeletal muscles, rigor mortis, and excitable tissue properties, resting membrane potential in a single muscle fiber, difference between single and multi-unit muscle fiber, isotonic and isometric contraction we're going to discuss. Then also we're going to discuss about excitation, contraction, coupling, and the skeletal muscles. So we're going to start with the difference between single unit and the smooth muscles and we're going to compare single unit with the multi-unit smooth muscles. So, for example, the single unit smooth muscles are present in the muscles of GIT, bronchi, urinary bladder, and in uterus. Whereas the multi-unit smooth muscles are present in ciliary muscles, muscles of iris, pilomotor muscles, in hair follicle. Then also the single unit muscle, the tissue maker are present, and here the tissue maker is responsible for the rhythmic contraction and relaxation of the muscle. Whereas in multi-unit, no such pacemaker tissue are found. Then another we have that in single unit smooth muscles, we have the autonomic nervous system that can modify the response, right? But in multi-unit smooth muscles, only show contraction as per the discharge in autonomic nerve supply. Then in single unit, the stretch of the muscles also causes the reflex contraction, whereas no effect of stretch on the muscles is found in multi-unit. Right. Then in single unit muscle, low resistance bridges are present in between the cell, so acting as a functional synectium. And in multi-unit muscles, no such bridges are found. Then in single smooth muscles, it contracts as a single unit and there is a widespread contraction. But in multi-unit, contraction is more discrete, fine, and localized. Okay, so these are the property of the single unit muscle and the multi-unit muscles, and based upon the location, all these action occurs. Okay, now going to start with the skeletal muscles. Okay, so if the question over the skeletal muscle will come, so the question can come like, what are the properties of skeletal muscles? What are the characteristics that is shown by the skeletal muscles? So we're going to discuss about some key points of the skeletal muscles, right? So any muscle mass or muscle tissue that is made up of large number of muscle cells or myocytes. So these muscles are commonly known as muscle fibers because of these cells are long and cylinder in appearance. Skeletal muscle cells are also multinucleated and they are arranged in parallel to one another, right? So they are arranged like this as shown in the picture also. They are arranged in a parallel, okay? So these skeletal muscle fibers are multinucleated. nucleated and arranged in parallel to one another. Now, each muscle cell or muscle fiber is cylindrical in shape, and talking about the average length of the each muscle fiber, so it is approximately 3 centimeter, right? And also it will vary from 1 to 4 centimeter. It will totally depend upon the location. Okay. Now, the diameter of the muscle fiber also varies from 10 to 100, right? So now we're going to discuss about some other structure that is also presented this skeletal muscles. Okay. Now, muscle fiber are basically attached to a tough cord of a connective tissue, and that is known as tendon, right? So this is the tendon. Okay, so muscle fiber are attached to a tough cord of the connective tissue that is known as tendon. And here tendon is in turn attached to the bone, and tendon of some muscles is thin, flat and stretched, but usually they are tough, right? Now, talking about the that other thing, that each muscle fiber is enclosed by a cell membrane, right? So this membrane here known as sarcolemma, right? And the cytoplasm is known as sarcoplasm. Now, various kind of organelles are embedded in this sarcoplasm, like nuclei is there, myofibril is there, Golgi apparatus is there, sarcoplasmic reticulum is there, then ribosomes are there, little lipid droplets are there, glycogen droplets are there. So multiple organelles are embedded in the sarcoplasm of the muscle, right? Okay, now each muscle fiber has also got one or more nuclei, right? In long muscle fiber, many nuclei are seen usually, and nuclei are oval or elongated and are situated beneath the sarcolemma. Right now, next we have the structure of myofibril. As we can see, basically, what is myofibril? The myofibril run through the entire length of the muscle fiber. In a cross section of the muscle fiber, the myofibril appear like a small distinct dot. Okay, so they appear like a very small distinct dot, and the diameter of the myofibril range from 0.2 to 2. Okay, and also its length vary from 1 to 4 centimeter. Now, talking about the sarcomere. What is sarcomere? As in the picture also, we can see the sarcomere. So, sarcomere is defined as the structure and function unit of skeletal muscles. Okay, so what is sarcomere? It is defined as the structural and functional unit of a skeletal muscle. Right? Now, as we can see in the diagram also, the sarcomere consists of one half light I band, then it has one dark band, or we can call it as an A band, then also it contains one half light band, and in the middle of the A band, there is a presence of H zone, and both the filaments, right, are connected with the Z line, as we can see here. Both the actin filament are connected with the Z line. This is all about the sarcomere. Then, if we talk about the electronic microscopic study of sarcomere, so we have the myofilament has two types. The first is actin filament, and second is the myosin filament. So what is actin filament? The actin filament are the thin filament with the diameter of 20 mu and the length is of 1 mu. Okay, and also these fibers extend from either of the Z line and run across the I band and enter into the A band up to the exome. Whereas the myosin filament are the thick filament. Always remember, these are the thick filament with a length around 1.5 mu, whereas the length of the actin filament is 1. Okay, so this is all about the filament of the the various myofilament type. Then if we talk about the composition of the skeletal muscles. So always remember that skeletal muscle has water. It has 75% water and solids are 25%. Right? In solids, it contains organic substances and also it contains inorganic substances. Right? So in organic substances, it includes various proteins like myosin, actin, tropomyosin, troponin, actinin, desmin. So about all those protein already we have talked in the last class. So it has proteins and other inorganic substances also it contains.
Share the screen now, ma'am.
Is it really visible? The screen is visible.
Yes, ma'am, visible.
Okay, now we're going to talk about the general properties. Okay, so it includes excitability, conductivity, liability, and contractility. Okay, so the excitability is the ability of tissue to react to the stimulation with change of the physiological properties and generation of process of excitation, and the excitability of cells and tissue is a basic function of. Okay, so whenever we talk about the excitability, so it is defined as the reaction or response of a tissue to irritation or stimulation. Okay, and also it is a physiochemical change, right? Then talking about the conductivity. So as the name suggests, conductivity means the ability of the tissue to conduct impulse is the very important property. With that, the tissues are able to conduct the impulse. Then next we have the liability. So liability means the ability of the tissue to conduct certain quantity of impulse per time. Whereas the last property and the very important property is the contractility. Contractility means the ability to change the size or to contract and produce tension. Okay. So here, the stimulus is a very important factor, so that we're going to discuss in detail. So what is stimulus? The stimulus is a change in environment. And also it is defined as an agent or influence or act which causes the response in a excitable tissue, right? So that's why the stimulus is considered as a very important component. Okay, so various types of stimulus are there. For example, mechanical stimulus, then electrical stimulus, then also we have thermal stimulus, and chemical stimulus. So as the name suggests, mechanical stimulus, right? So mechanical stimulus means any kind of pinching, right? Pinching that is produced to make a change in the environment. Then, as the name suggests, electrical stimulus. Electrical stimulus means any kind of electric shock that produce a change, right? Then thermal stimulus, for example, any heat, glass rod, or ice piece that is keep on changing the temperature, and that's why there will be a change inside the environment. And the last we have the chemicals. So as the name suggests, any chemical substance like acid, due to which it generates a particular type of change. Okay, now there are some quality of stimulus is also there. Like to excite a tissue, the stimulus must possess two characteristics. Like first we have the intensity or strength, and second we have the duration. Okay, so talking about the intensity, so intensity, depending upon the intensity or strength, stimulus can be categorized into some minimal stimulus, some maximal stimulus, and supramaximal stimulus. Okay, so if we talk about the minimal stimulus, so means it has a strength or voltage that is sufficient to excite the tissue, right? Supramaximal means the maximum stimulus can be applied to produce a change or to excite the tissue, right? Then duration, like whatever may be the strength of the stimulus, it must be applied for a minimum duration to excite the tissue, right? And the duration of the stimulus depend upon the strength of the stimulus, right? So this is all about the stimulus. Then next we have the difference between isotonic and isometric. Okay, now muscular contraction usually classified into two types on the based on the change in the muscle of change in the length of the muscle fiber, and also depending upon the tension. Okay, so depending upon the length and the tension, we classify isotonic and isometric. Okay, just for example, if we talk about the isometric, right? So isometric is a type of contraction, is a type of muscular contraction in which the length of the muscle fiber remains same and the tension is increased, right? So muscle contracts but does not shorten. Okay, just for example, in pulling any heavy object, when muscle becomes stiff and straightened with increased tension, but the length does not change. Okay, now talking about the tonic, isotonic. So as the name suggests, tonic. So here what will happen? This is a type of muscular contraction in which the tension is going to remain same and the length of the muscle is going to alter. So as we can see in this slide also, the length will be increased, but the tension is going to remain same. Okay. So the example could be simple in the flexion, any flexion of the where shortening of the muscle fiber will occur, but the tension is going to remain same, right? So if we talk about the difference, so we can write that in isotonic, what will happen? The tension remain same, the length of muscle fiber is altered, right? And the example just now we have discussed the flexion of arm, right? Then if you talk about isometric, so in this, what will happen? The length of muscle fiber remain same and there will be increase in tension. Okay. And just now we have discussed the very example like pulling any heavy object. So student, please remember that the question over the difference between isotonic and isometric can become. Okay. Then next we have the trigger mortis. So what happens usually in this case, this rigor mortis concept is basically, this is a condition to the body after the death, right? So this is characterized by the stiffness of the muscle and the joints, okay? And this is occurred due to the stoppage of aerobic respiration, which causes the changes in the muscle, okay? Now, first we're going to know the concept. What is rigor mortis? This is the condition of the body which occurs after death. And this is characterized by stiffness of muscles and joints, right? And why this occur? This occurs due to stoppage of aerobic respiration, which causes the changes in the muscles. Okay. Now, we're going to discuss about the causes of this rigor mortis. So what happens soon after the death, as the cell membrane become highly permeable to calcium. Okay, after the death, the cell membrane become highly permeable to calcium. So what will happen? A very large amount of calcium is going to enter inside the muscle fiber and promote the formation of actinomyosin complex, which results in the contraction of the muscle. So as we know, the calcium is a very important component for the muscle contraction. So after the death, there will be permeability of calcium, which causes the muscle contraction, which causes the formation of actinomyosin complex, right? Now, what happened? A few hours after death, all the muscle of the body undergo severe contraction and also become rigid, right? And the joints also become very stiff and long. Okay, so after the some hours, these all the muscles, because they are keep on going on contraction, so they become more stiff and rigid. Now, what will happen? Normally, we know for the relaxation, the muscles need to drive out calcium, which requires ATP. But during continuous muscular contraction and other cellular processes after death, the AP molecule get exhausted. Okay, so as we can see here in the picture also, there are no ATP molecules are present after death, and this slide is basically indicating the condition after death. Okay, so as you can see, during normal condition, the ATP molecule are present, but after that, the ATP molecule get completely exhausted, right? So, new ATP molecules cannot be produced because of the lack of the oxygen. So, in the absence of ATP, the muscles remain in the contracted state until the onset of decomposition. Right now, why this rigor mortis is very, very important. Usually this is utilized, or this is very useful in determining the time of death. Okay, so onset of stiffness start between 10 minutes and between 10 minutes to 3 hours after the death, depending upon the condition of the body. And also, it will depend upon the environmental changes that occur with the body, right? And if the body is active or the environmental temperature is high at the time of death, the stiffness will occur very, very quickly. Okay, so this concept is very useful in determining the time of death. Okay, now what happened afterwards? The decomposition of the general tissue starts, and the lysosomal intracellular hydrolytic enzymes, various enzymes are there, as we know. They will release, and the enzyme hydrolyze the protein, the muscle protein, which are responsible for the contraction. Now what will happen? It relieves the stiffness of the muscle. Okay, so this is all about this concept. Now next we have the fatigue. What is fatigue in skeletal muscles that we're going to discuss? So as we know, what is fatigue? Fatigue is defined as the decrease in muscular activity due to repeated stimuli. As we can see, from a single point of stimulus, repeated uh, muscular activity is occurring, right? So what is fatigue? It is a decrease in muscular activity due to repeated stimuli. And when the stimuli is apply repeatedly, what happens after some time? The muscle does not show any kind of response to the stimulus, and that condition is known as fatigue. Right now, as we can see in the curve also, when the effect of the repeated stimuli is recorded continuously, the amplitude of first 2 or 3 contraction is increased. So as we can see, this is first, second, and third. So it is keep on increasing, right? Now, why it is occurring? This is due to beneficial effect. This is due to beneficial effect, and afterwards the force of contraction will decreases gradually. So in the 10th, in the 30th, 40th, it is keep on decreasing, right? So it is shown by the gradual decrease in the amplitude of the curve, as we can see. Right? So it keep on decreasing, and the amplitude is also keep on decreasing. Now, all the periods are gradually prolonged, and just before fatigue occurs, the muscle does not relax completely. It remains in a partially contracted state. As we can see, it remains in a partially contracted state. And this state is known as contracture or contraction reminder. This state is known as contraction reminder. Okay. Now, what are the different causes that is responsible for fatigue that we're going to discuss? So, causes of fatigue we have the exhaustion of acetylcholine in motor weight. Another could be lack of nutrients like glycogen. Then the lack of oxygen is the another reason, and it could be also occur due to accumulation of metabolic like lactic acid. So these are the reasons for the fatigue. Right, so this is all about the curve of the fatigue. Okay, so next we have the RMP in a single muscle fiber, RMP or the resting membrane potential in a single muscle fiber that we're going to learn now. Okay, now what is RMP? Resting membrane potential is defined as the electrical potential difference or the voltage across the cell membrane. Right? Now, this resting membrane potential is also known as trans membrane potential. Okay, now what happens when two electrodes are connected to a cathode ray oscillator through a suitable amplifier and played over the surface of the muscle, there is no potential difference is noted, that is zero potential difference. Okay, but if one of the electrode is inserted to the interior of the muscle fiber, the potential difference is observed and across the sarcolemma, and there is a negative inside and positive outside. So as we can see, there is negative inside and positive outside. Okay. Now, exactly what is resting membrane potential is defined. So as we have discussed, the resting membrane potential is the electrical potential difference across the cell membrane in resting condition, and in resting most of the cell have minimum or nil permeability for the sodium and calcium, right? But one ion is very freely movable in and out of the cell, and that is known as potassium. Okay, so the development and maintenance of RMP depend mostly on equilibrium potential of potassium. Okay, now we're going to discuss the RMP in different tissues. Okay, so if it will be different in every tissue. For example, in neuron, it will be 70 minus 70 millivolt. In skeletal muscles, it will be minus 90. In cardiac muscle, it will be minus 85. Then in smooth muscle, it will be minus 52 minus 95. And also in RBC, it is minus 12 milli. Okay, so change in RMP mostly depend upon the movement of ions, anions and cations, right, in and out of the cell membrane. And in resting condition, as we can see, what happens in resting condition, there will be more negative inside and more positive outside. So in resting condition, anions are more in intracellular fluid and the cations are more in extracellular fluid. Okay, and anions, as we know, anions are negatively charged and cations are positively charged, right? So their movement will occur in opposite direction through the cell membrane. What will happen? The exit of an ion from the cell into the ECF will produce the depolarization. In the last class, I think we already have discussed that what happened when there will be exit of these anions out of the cell, that what will happen, the process of depolarization will occur, and the entrance of an ion into the cell from the ECF will cause the hyperpolarization, and that will generate the action potential. Okay. So if we talk about the curve, so what will happen? If this is the resting membrane potential, that is minus 70 MV. So from here, there will be production of depolarization, and after reaching a threshold, then this will cause the hyperpolarization. It will be like this. Okay, so this is all about the curve. Okay. This is the depolarization, and this is hyperpolarization. Okay. Okay. So this is all about the RMP. Now we're going to discuss about the excitation contraction coupling. Okay, and I think some part of it already we have covered in the last class. So what happened, as we have learned, the muscle fibers have two tubules, the L tubules and the T tubules. Already we can see in the diagram. So T tubules are basically responsible for the formation of the sarcoplasmic reticulum, where they stores calcium. Okay, so what happened whenever there is excitation of the muscle fiber by impulsing through the motor nerve and neuromuscular junction? So what will happen? This will causes the spread of the potential along the through the T tubule, right? Now what will happen? There will be entrance of calcium in cistern, and there will be release of calcium, which causes this calcium is going to bind over the troponin, and troponin has already binded to the actin filament at the active site. And as there will be removal of the tropomyosin, then this myosin is going to bind. Okay, so for better understanding, we're going to draw a quick chart. Okay, so excitation of muscle fiber by impulse passing through motor nerve and NMJ. NMJ already we have covered in the last class. So if you have any doubt over the NMJ, so you can watch the last class. Then there will be production or the generation of action potential in the muscle fiber. Then it will cause the spread of action potential through T tubule. So it will cause the entrance of action potential, which will cause the release of calcium ions from T tube. Now this calcium move towards the actin filament and will bind over the troponin, and troponin will get removed from the active side of the actin filament where the myosin can bind, and finally it will cause the muscular contraction. So this is all about the excitation coupling reaction. So I hope all the students find this class more useful, and already we have covered all the topics that was shown here. We have completed fatigue, rigor mortis we have completed, excitable tissue property we have done, RMP, difference between single and multi-unit muscle fibers, isotonic, isometric, excitation, contraction, coupling reaction, and also the features of these skeletal muscles we have covered. And these are the important topic of this unit, and also some important topic already we have covered in the last class. So if you have not watched that class, you can go back and also watch that class. Okay. So various important topic like NMJ, sliding filament theory, the difference between smooth muscles, skeletal muscles, cardiac muscles we have covered, the various properties of the muscles already we have covered in the last class. Okay, so this is all about today's class. And if you have any query, any doubt, so you can ask or reach us to these particular platforms. Okay, so thank you so much for watching my class. Durga Ma.